Hey class, in this recording we're going to be focusing on fetal development. So in our last series of recordings, we focused on changes that happens into the adult female's anatomy during pregnancy and childbirth. During this series of recordings, we are going to focus on changes that happen to the embryo and fetus, or the kiddo, during this developmental process. So let's define some terms here. As we look at an embryo, this term can have a lot of meanings to a lot of different individuals. When we think of what an embryo is, to some people, an embryo is just anything that's at the fertilized egg stage or beyond. Other individuals think that we need to wait until at least 16 days to get to that embryo stage because we have the ectoderm, mesoderm, and ectoderms. They want to have those 3 germ layers. And for these individuals, they call the stage before there are the 3 germ layers the pre-embryonic stage. And you'll notice this the longer you've been in academia. There's always going to be more terminology added, not less. And as we look at our lecture textbook, our lecture text takes a pre-embryonic stage approach for before 16 days and then an embryo after 16 days approach because we're basing our lecture presentations off of the Saladin text. So as we look at this process of getting sperm into the reproductive tract, we need to get those eggs fertilized within 12 to 24 hours after ovulation. And as those eggs are moving through the reproductive tract, they need to be caught by the fimbriae of the fallopian tube or uterine tube, and it's usually right about there, that distal 1/3, usually at an interface between the ampulla and infundibulum of the fallopian tube or uterine tube, where fertilization is going to occur. And once that fertilization occurs, that egg is then going to be recategorized as a zygote. Now, we need to really emphasize that the overwhelming majority of sperm do not make it to the egg. Right off the bat, a gajillion sperm are going to be destroyed by acid within the vagina, or will just ooze out of the vagina as the fibrinolysin will remove the coagulatory proteins. A lot of the sperm also are going to fail to penetrate the mucus of the cervical canal. So that thick mucus within the cervix prevents a lot of sperm from getting in. And then within the uterus, there are white blood cells roaming around that will phagocytize sperm and destroy them. And then there's 2 uterine tubes, and half the sperm, they take a wrong turn and they end up at the wrong destination. They go up the uterine tube that, well, doesn't have an oocyte to fertilize. So of the approximately 300 million-ish sperm that are ejaculated, only about 200 spermatozoa or sperm get anywhere even close to the egg, the general vicinity of the egg. And as we look at the sperm, they can hang out in the female digestive tract for up to 6 to 7 days. So I just like to say a week and play it safe. And this is important from a family planning perspective. You really need to know, okay, if I'm going to be ovulating on this day and I have sexual, that means that these are the days where I can or cannot have sexual intercourse in order to make sure that I don't have viable sperm within the female reproductive tract. Well, there is a viable egg. So as we look at conception, this process of conception is going to involve a sperm getting to the oocyte. And usually conception is going to be optimal if we give the sperm a head start. So we give the sperm time to swim through the reproductive tract of the female's body. So in other words, if the woman has sex a couple days before she ovulates, or up to 14 hours after ovulation occurs, because it takes some time for the sperm to get there. And when that sperm encounters the egg, that sperm is going to release enzymes from the acrosome. And that acrosome is right at the tip of the sperm and contains a lot of digestive enzymes. And those digestive enzymes are going to be used to burrow through the granulosa cells that make up the corona radiata around that egg. And then once they get through the granulosa cells, there's this membrane known as the zona pellucida that the sperm also needs to burrow and digest its way through, until finally we have the fusion of the sperm with the egg, and the sperm nucleus can be released into the egg to fertilize the egg. Now, this process of fertilization is going to involve taking one set of DNA, a haploid genome from the sperm, and the haploid set from the mother, or the egg, and combining them to make a diploid set. So recall, we went through meiosis, where we took a haploid germ cell, we took a diploid germ cell and turned it into a haploid gamete, and then we take the sperm, the male gamete, and the egg, the female gamete, and combine them together. So we cut the number of chromosomes in half to combine them again and get back to baseline. And we don't want extra sperm getting into that ovulated egg, because if we have too many sperm get in, we'll have too many chromosomes. We'll have polyspermy, basically result in this being a non-viable pregnancy. The embryo will not make it to the fetus stage. And there's two ways that we keep extra sperm out. We have the fast block and slow block. That fast block channel is going to involve sodium-gated channels rapidly opening in the egg membrane, and that's going to prevent the sperm from being able to penetrate by changing the voltage across the membrane of the egg. And we have a slow block method. And the slow block method, I'm going to have to back up a slide here, is going to involve some secretory vesicles just beneath the surface of the membrane. You can see those secretory vesicles shown here as green dots. And once the sperm penetrates, those green dots release their enzymes, and they make the membrane of the egg thick and impenetrable to the sperm, or I should say subsequent sperm that try to get in, so that we can prevent polyspermy from occurring. Now, as we look at the secondary oocyte, or egg, as I've been just referring to it as, that secondary oocyte has been frozen in meiosis 2. And it's only going to finish meiosis 2 only if it's fertilized with a sperm. So as we're going through this process of meiosis, we need to have a second polar body ejected from the oocyte. And that second oo body is going to be ejected as it's removing just a little bit of extra DNA material. And this polar body, you can think of it just as genetic leftovers at this point in time. And it can do this by removing one chromatid from each of the chromosomes. So as we look at these replicated chromosomes, your classic replicated chromosome, we have a chromatid on the left and a chromatid on the right. And by removing one chromatid, we are then going to have removed a copy of DNA. So now we have a sperm and an egg that have combined. Their nuclei swell together, or swell and become pronuclei. And at this pronuclei stage, eventually they pop. You can think of these as being like overinflated balloons. And the chromosomes from the father and the mother, the paternal and maternal chromosomes, are going to mingle to form one diploid set of DNA. And now at this point in time, boom, bada bing, bada boom, we now have a unique human genome that has been formed. And this brand new, unique human genome is going to be in the fertilized egg. We have a single cell with a brand new human genome, and we call that single cell a zygote. And now that zygote is going to go through many, many, many rounds of cellular division. And eventually, the goal is to have that zygote end up as a baby that could grow up to be a whole, a functional member of society. So that's all we have for this recording. If you have any questions or comments, please feel free to let me know and post them in the discussion boards. And as always, happy studies.